So today we'll be talking about some new RNA-Seq analysis tools, and so we'll go through an assembly and some of the downstream analysis that will occur in our RNA-STAR and GenVision Pro. And specifically, we have some new tools for isoform analysis. Before we get started on the live demo, though, I'd like to give some background information regarding DNASTAR. DNASTAR was founded by Dr. Fred Blattner. Dr. Blattner sequenced the E. coli genome in the 1990s as one of the genomic founders. He co-founded NimbleGen, which is now Roche, and he's also the founder of Scarab Genomics and DNASTAR. And Dr. Blattner continues to be the head of development for DNASTAR, and we're developing many new things, some of which I'll mention a little bit later in the webinar. So really the goal for DNASTAR is to design software that is easy to use and also very powerful. So it's a combination of power and ease of use that makes us popular for a wide variety of different sequence analysis types. And so one of those is molecular biology. We have a suite of software that we call the Molecular Biology Suite that offers many different types of analysis tools. And I just have some screenshots here. You can see there's some multiple sequence alignment. So we can align DNA and protein and look at things like trees and multiple alignment diagrams. We also have Sanger sequence aligner and editor. That's very good. And we have some new tools for auto annotation. About a month ago, we had an auto annotation for plasmids added to our Seq Builder program, and it provides a really nice interface then for adding annotations and accepting them. So it's really a wide-ranging set of tools for molecular biology, and this is, again, we have separate webinars that cover all those specific topics. Today we're going to be focused mostly on the Genomics Suite, and the Genomics Suite has support for a wide range of different workflows. And so it can range anywhere from genomic workflows to transcriptomic, de novo workflows, templated workflows, as well as other types of projects like virus analysis or metagenomics. So it's really wide-ranging, and we provide both the assembly algorithms, and they're easy to set up, as well as all the downstream analysis that you'd want. So we're going to start with the assembly software and then focus on some of these new analysis tools, and you can see just some screenshots here of what some of those tools look like. And then we also have structural biology, so some brand new software that's been developed just in the last few years here at DNASTAR that gives you some really excellent visualization of proteins and does protein prediction and can be used for things like antibody design. So these are, again, we have a separate series of webinars and videos that cover topics in structural biology. So taken all together, the molecular biology, the genomics, the structural biology suite is the DNASTAR suite of software. So it's really wide-ranging functionality that we're offering. So again, the vision is to provide, all the basic molecular biology software tools. So it's a whole suite of programs that provides, cloning, primer design, sequence editing, annotation. And then we have our NGS tools where we can take large, complicated data sets, easily align them and analyze them, both for transcriptomes and genomes. We have cutting-edge protein tools. And then looking ahead to the future, we're also focused on long-read sequence technologies. So later this year, we'll have probably some more webinars and some more discussion about, some of the new nanopore data that's hitting the market. We have clients worldwide in academia and the government, biotech, pharmaceutical. And then we also offer support. And our support is we have chat support, webinar support, email, phone calls, so we cover a wide range of hours, as well as we have different global locations. So, and support really distinguishes us from many of the competitors. So if you have technical questions, you can always reach a DNASTAR representative that can, help you with your project. So one of the focal points in the last couple of years has been accuracy, and we've got lots of documentation on getting our NGS assembly software to be as accurate as is possible. And And we did that using some publicly available data sets. And this is from the NIH, the Genome in a Bottle Consortium, that sequence a human genome. And actually now it's more than one. But they sequenced them using multiple different techniques and then validated that variations are present. And we can use that as essentially the answer key to take our software, to take that same data, assemble it, check the accuracy of the assembler, then filter the variants, and we can test the filtering accuracy, and then at the end result, do we find all the variations in this human genome that are known to be there? And so we can determine very precise accuracy measurements, and we're, we think in a very good position, over 99% accurate. And when we compare ourselves to some of the other assemblers and SNP callers, one of the most common, of course, is the BWA mapper and the GATK SNP caller, as well as a couple of commercial competitors. We find that, if we compare false positive detection rate and true positive detection rate, all which contribute to the overall sensitivity, that we are the most accurate. And we're also very fast, so we can do both the assembly and the SNP calling. And in this case, it's an exome in a little bit over an hour. In some cases, now with the later builds of the software, under an hour. Whereas our competitors really struggle with things like false positives can be quite high, so this FP column. And we also do a very good job with the false negatives. So those two things together give us the highest degree of accuracy. So speed is another component, not as important as accuracy, but speed, of course, is, especially if you're doing a lot of samples, you want to make sure that you're using something that's as fast as possible. And so we have some other benchmarks on a whole bunch of different data sets. And I just kind of lightly highlighted what we're going to be looking at today, and that's some RNA-Seq, some universal human reference and brain RNA-Seqs. And, they take about anywhere between an hour and an hour and a half. Of course, you may have 50 samples that you want to do. So if you're doing large-scale RNA-Seq, that speed starts to matter a little bit more. It helps if it takes just an hour versus 6 hours per sample, because it means the difference between running assemblies for a day or running them for a week. So we think we're in a really good spot in terms of our overall accuracy and our overall performance in terms of speed. And so at this point, we're going to focus on one of these RNA-Seq data sets, and we'll show you the project setup and how easy it is to set up those assemblies, and then we'll, again, explore the downstream analysis and do some focus on some of the new isoform analysis that we've added. So I'll exit our PowerPoint here and folder. So our starting point is the assembly software, and this is SeqMan Engine, and it really is a project setup wizard. So it allows me to pick the type of project that I want to do. We're going to assemble this locally, and we choose the workflow. In this case, it's a transcriptomic workflow, and it will be RNA-Seq. And we just click Next. And you can see this side panel as we progress through the wizard. This will start to build. And I can go back by clicking these buttons and move from page to page to see where I've been. And then we choose the assembly type. And, of course, with transcriptomics, we have reference-based, and we also have the option for de novo, two totally different sets of algorithms. So they have slightly different hardware requirements, and they're quite different in the algorithmic approach. But for isoform analysis, we're going to focus on alignment of that sequence data to a reference genome. And using that reference genome, which, of course, has intronic regions and exons annotated, we should be able to determine, in some cases, when there's alternate exon usage. We collect some system information, and there's a whole link here to technical requirements that will give you more specific info. And that reference genome, we can get it from multiple different places, and this is one of the what makes DNASTAR software so powerful is that we can work with model organisms and non-model organisms. You can load in all different types of reference sequences. One place I can just go to is NCBI. So we have a link here that goes to NCBI, and this is relatively new in the software. And I can pick whether it's a eukaryote or a bacteria, and then from this pull down, I can select from all the different genomes at NCBI. So it makes it convenient then to go and download all the chromosomes from an organism, which is surprisingly difficult going to NCBI directly and trying to download genomes. So we make that a little bit easier. We also have genome template packages, And genome template packages are a combination of the annotated reference genome plus the DB-SNP database, if there is one for that organism. And there's a relatively small group of organisms, of model organisms here. We have several different varieties for human, where both of those are available. And so those you can just download. Our customers can go and automatically download those into their assembly. And I have one already downloaded. This is human build, genome build 37 with the most recent DB-SNP build 146.genome template. So that's what's in that package. So I'm going to use that to align my RNA-seq data against it. Now I can also pick, when we do RNA-seq, we want to quantitate expression against the annotations that are on the genome. So we have an interface here that allows me to pick the subset of features that I want to annotate against. So some cases, many cases, it's just the CDS sequences. Some genomes, though, are annotated differently, and it's genes and mRNAs that I want to quantitate against. So you have some selection. I might decide I don't want to quantitate ribosomal RNAs. So you can select what you want to quantitate against, move on to the next step, which is load the sequence data. So here, of course, we select from our pull down our type of our sequence data that we're loading, and I'm going to load in some, and this is, again, some of the reference RNA-seq data that you see at many different locations, and I'm just going to load in some brain, and I'll do that again, and universal human reference. And so I have two samples now, and so I tell the program that we have two samples. I'm going to expand this window a little bit so I can read the label, and now I'm going to just group the brain. And if you have replicates, it's the same kind of a process. I would name each replicate and then group them by experiment. In this case, I'm just going to set it up like how our rest of our demo is going to go, and that's not with replicates in this case, but just brain. So I'm just selecting here and grouping them. And there's different ways to do this as well. I can auto-name. If my files are named in a certain way, it will grab the file name and auto-name them. So there's a couple different ways to automate this. So now I have a 2-experiment RNA-seq, two different tissue types, with 8 different files. So quite a bit of data, but certainly reasonable for RNA-seq. You could have quite a bit more data than this per experiment. And one of the common questions is, well, how much data do you need to do RNA-seq? And it really depends on how much sequence coverage you need for the genes that you're interested in. So if you're primarily looking at genes that are expressed highly or moderately, 50 million reads might be sufficient, maybe even less than that. If you're interested in genes that are expressed in the low end or expressed at low levels, then really the more data you can add, the better. And because all the RNA-seq is going to be expression values, and the isoform analysis is going to be based on depth of coverage. So you need to have coverage across those genes that are expressed in the low range if you want to analyze those. So that's just something to keep in mind. More data is usually better for RNA-seq. So then we have some basic options. Sometimes with RNA-seq, there's adapters or linkers present. I use a free program from the Babraham Institute to scan files. If I have new files I'm not familiar with. And it's not too uncommon to get things like Illumina universal adapters present. And if you can identify that, you can run this scanner and it'll trim that out of the sequences. It's not actually necessary, though, for a templated assembly. Our assembler is very robust. If there's portions of the reads that don't align due to linkers, it's very good at trimming them off, but it doesn't hurt sometimes to trim some of those off prior. So you could turn that on if you wanted to. The one parameter here, assembly-wise, that you might change is the minimum match percentage. If I make this number lower, it allows for more mismatches. In this case, 85% means up to 15 mismatches per 100 base pairs. And that will allow, especially if you've sequenced, say, a strain, or your reference isn't the exact same genomic strain as what you've sequenced, you might lower this match percentage a little bit to allow for more mismatch, but we'll just stick with defaults. Now, our software can also do multiple types of analysis. So I can also do SNP analysis at the same time that I'm doing gene expression and isoform analysis. So if I turn on the SNP caller, I can do that. If I have a human genome, I can also import variant annotation information. By checking this box, it'll go up to a cloud, pull huge amounts of data into my project, and these are things like clinical information on particular SNP positions or functional predictions on particular SNP positions. So if you want to do SNP analysis and expression analysis, you have that available to you. The normalization we have set to RPKM, which is fairly typical. And there's some advanced options here as well. One thing I just wanted to point out is that with RNA-Seq, depending on how your data has been prepared, you may or may not have some extremely abundant transcripts present. And so those can be problematic if, as they start to get to the hundreds of thousands or millions of X coverage, especially if you're doing SNP calling. And it might just slow things down. So there is some ability to limit those ultra deep areas. By default, we'll limit coverage to mitochondrial or chloroplast genomic templates if they're present. So that's just something that helps make the assemblies go faster because it'll limit these areas that can be extremely deep in RNA-Seq. But you can turn that off too and just say, no, I want full coverage on these organelles. As well. So it's just something to keep in mind. And sometimes you might think that your ribosomal has been depleted in your sample, and you find out that it isn't. And sometimes the way you find out is when the assembly takes much longer for one of the samples, and it turns out that 80% of your data is trying to align to a couple of genes. So that's just something that we can help you with too, and troubleshoot if you run into those issues. We can set a gender. This is for SNP calling, and then we just name our project. And you'll see that the output files then, so I'll just save this to my demo data folder. We have an A star and we have some dot assemblies. The A star is an array star project that allows us then to compare and find now those genes of interest based off expression values or isoform values. We also get the assembled data. These dot assemblies contain all the BAM files, one for each chromosome. That allows me to go and look at the aligned data and look at SNPs and look at each read as it aligns to the genome. So we've got a couple different outputs. We can also select wiggle files. So a wiggle file, of course, typically is a coverage file. And you can use these wiggle files produced here if you want to do some analysis, say, in a genome browser. And I'm going to have it average every 5 bases. So if I want to take these coverage maps and bring them elsewhere, wiggle files is a good thing to save out as well. And then everything's ready to assemble. When I start the assembly, we can watch the progression. When it's done, there will be a button here that says, launch in SeqMan or launch in ArrayStar, GenVision Pro. And typically, we'll go right to ArrayStar as the typical next point. So I'm not going to run the assembly here. I'm just going to quit. But when this is done, this page will change. And we'll just diminish this. And I'll go to the ArrayStar project. And so ArrayStar has, there's experiments now. This is a very simple RNA-Seq where I have brain and UHR. And then we also have the variation information set up as separate experiments as well. So they're separated in ArrayStar. We're going to focus on the expression for this webinar with the brain and UHR. And usually the next place I like to go is just the scatter plot. And the scatter plot then is going to compare UHR, in this case on the y-axis, and brain on the x. And because this is two completely different tissue types, the correlation isn't really tight. We have kind of this more spread out pattern. We have much different, both in isoforms and in gene expression per gene, as you would expect. If you have all one tissue type, these scatter plots will be very, very tight in some cases, with very few genes that show differential expression. So in ArrayStar, there's multiple different things that I can do. When I look at a scatter plot like this, there's a lot of different genes here, right? So the object now is how do I go from all the genes to find just those that I'm interested in? And so that's really what ArrayStar is for. Is it takes in whether it's gene expression or SNP variation information or ChIP-seq data. It takes lots of data points in and provides you with a whole set of different types of filters narrow this down to something that's manageable and interesting. And so ArrayStar has extensive filtering tools. So if I go under the filtering menu, I can see that, oh, this project had some filtering applied, and I'll kind of go through. I can filter the gene table. I can also filter the variant table. So if I want to look through variant analysis, and I can also search at the exon and isoform level. We'll focus on the gene and isoform tables and set up some filters that way. And so the way that I'm filtering this now is I'm filtering the gene table, and I'm just saying from this pull down, show me just genes that have a fold change between the two, and I can set this fairly high as well. Excuse me. So either experiment, show me all those genes that have fold change of 8-fold or higher. And actually, I'm going to back up to one other step here. So another thing I can do is I can say I'm really only interested in things that are expressed at a fairly high level in this case. So I can kind of combine. I can directly select in the scatter plot. And so that narrows this down to about 5,000. Let's grab a few more genes. So there's about 6,000 genes that are expressed fairly high between these two tissue types. Now I'm going to say, instead of filter all, I'm going to say filter just that selected group with this set of filters. And the reason that I'm doing that is I'm going to set a cutoff. I don't want to analyze genes that are at the low end of the range where there's just a few reads aligned. It's going to be very difficult to do isoform analysis when there's actually not coverage across the transcript with sequence data. So it really limits your ability. So you have to kind of focus. If you're looking for rare isoforms or exons that are expressed alternatively, you're typically going to find those most consistently when there's enough expression and enough sequence data. So I'm going to focus on a fold change then between these two. And then I'm going to add another filter, and that's gene annotation. So I can add additional filtering criteria in ArrayStar. And from the pull down, I can filter from the expression, the sample group, the annotations that are present, fold changes, ontology, or gene classification, depending on what I brought into the project. Now, in this particular project, I imported some additional annotation information. So I can do a double filter, actually a triple filter, highly expressed, fold change, and now I just said in the annotations, look for the word cancer somewhere, and then I can search. And here I end up at the bottom. There's 51 genes then that have a dramatic change in fold change, and somewhere in the annotation have cancer. And so I've narrowed this list down to something much, much smaller. And here's just a list of the genes then that meet that criteria. And you can see that in this table, I have some columns of RPKM for brain. So this is a log 2 scale. And then I have UHR, and then I have disease description, function description, trait association. The way that I imported these is I did a file, import annotations, and I imported DB-NSFP gene, which is a set of annotations that's publicly available. We have it on our website as well. Actually, we probably have a newer version that has all different clinical and functional gene annotations. But you might have your own annotations, and you can see that I can bring in a wide variety of formats to populate this gene table, and then I can filter on those terms. So if you have your, an Excel sheet that has your genes in it and descriptions, those can be imported through a custom import wizard. So there's multiple different ways to pull in information into ArrayStar. There's also some automatic connections to some databases, so I can download, here's a Homo sapiens Go term, so this is more ontology type annotation, so you can bring that in as well. So there's just multiple sources that I can import from. So however you do the filtering and you've narrowed down the gene set, now at this point I can start to do some additional analysis. So I might say, well, I'm just interested in, looking at all these genes. I want to visualize them and see, well, I know there's expression differences. Is there any observable isoform difference in them? And so I can select all of them, and then I'm just right-clicking now, and you can see I can send the selection to GenVision Pro, or I can send it to SeqMan Pro. GenVision Pro is a newer application, and it's a genome browser that allows me to look at tracks of data. So I can have multiple different experiments. I can look at both coverage tracks and also sashimi plots that show me the different isoforms. If I want to look at the raw assembled data and the reads, I can go right to SeqMan Pro and look at specific spots in the assembly. So when I send a GenVision Pro, I can send a selection of up to about 100 genes at a time. In this case, I have 51, and I've done that already. It takes just a few seconds to do it, but I've already loaded it up. And so GenVision Pro will automatically open to the first, in this case, the first gene on the list, which is this T-span7 gene on chromosome X. And it will automatically open that up. And right now it's loading some additional files. So in this particular project, I've loaded that chromosome X. It's navigated right to that position that I want to analyze, and I can see these sashimi plots with the arcs. And I'm just going to change some options here. So I'm selecting those tracks, and you can see under the options I can change the color of arcs. So I can really make this now look, and we'll zoom in at different levels too. But I have arc color and coverage color and background, so I have control over it. I can also say the arcs then are the reads that split between adjoining exons or between non-adjoining exons if there's some alternate exon usage going on. So I can control what this picture looks like. I can say, put a minimum read count. If I completely remove the arc count, so we'll go to 1 here, and you'll see, oops, that was 110. We'll try 1. So there's all the filters gone. You can see we get all these kind of extra spurious arcs, and that's real data, but it's probably not very interesting data. There's oftentimes some background noise in the assemblies. So I have some control then over my arc filtering. I also have some control over how far I should look for the span between these reads that are split between adjoining exons, and that's what the maximum span is. And I can change this number as well, and I can make it if I make it a really big number, something with my webinar is fighting me on this window, but I'm going to have to see if I can get it to go here. No, we're having a WebEx struggle for my window here. Let me see if I can get this to add. No, I can't with the WebEx running. Unfortunately, I can't click that. Sometimes the webinar will block a certain box like that, but I can change that number. I had already changed it to a fairly big number of 120,000, and that's to support this particular exon you can see here that is about, well, it's about 100,000 span between exon 1 and 2 for this particular gene. And so there are some genes that have even a greater span. If I make the arc length too big, there may be some spurious. Again, pseudogenes that are in the neighborhood, there may be some reads that split between gene and pseudogene. So we can kind of filter that out as well. I can also change the axes. So here it's set, well, we'll try to change it with the webinar. We'll see if it'll let me do that. But so the axes is set to 5,000, and nope, it won't let me change these with the WebEx clone, but I can change the scale. So we're at 5,000 here. Some genes can reset this to 500 or 100 and see that better. Now I'm going to zoom in a little bit, and you can see that we can, if we see an area that's interesting, we can move and now look at these coverage. These sashimi plots have coverage plots, and then the arc with the numbers, the number of reads that split between those exons. In this case, we don't see anything. It looks like the pattern, you know, the isoforms look pretty much the same between brain and UHR. And what I have below here, I've also imported wiggle files. And so you might have wiggle files from another source. You want to compare them to what you just assembled in DNASTAR. I can click this button, import wiggle files into the project. If you don't need to look at wiggle files, I can go here to the controller that is going to control what's visible in the screen here, and I can turn off those wiggle files. And of course, I can resize things and move things around. So if you want more real estate for viewing your tracks, you can change that. I can also change the layout. So I can change the space before the tracks. I can change the height of the tracks. All right, space after it. So I've got lots of flexibility here to change how this sashimi plot looks. And then if I want to look at, here's the list then. So what I just sent was this list. So here's the next gene, BEX2, that was on the list. And I can see, oh, on this one, it was expressed quite a bit higher in brain. You can see from the coverage. And it looks like there's some alternate splicing possibly going on between, in this case, exon 1 and exon 2. So it looks like the brain has higher expression. Maybe some indication here we've got a couple of isoforms that are present. And I can look at the annotations now to confirm, is there an isoform? And here, it's a little hard to see this one, but you can see that if I hover over the annotation, that this is transcript variant 4, and it looks like with transcript variant 4, exon 1 is a little bit shorter. If I put my selector over that, I can see, oh, that's what these arcs are, 34 and 346. Most of the isoforms are this transcript variant 3, but it looks like there's a small subset belong to this other transcript. So you get this nice picture then of the splicing. Then we get these other little arcs like this 12. I don't know that that's real. I don't see any indication in the annotations. Oh, there's something as I scroll up. Here's these other isoforms then. If I hover over it, I can see an isoform 2 that starts right at that same spot. So I can visualize now at the 5' end of these transcripts that looks like we have 3 different isoforms present. And it's very efficient now. I can go through this list, and it's just a great tool. I can zoom in and out. Focus on areas of interest. So here's kind of a classic form where it looks like we're skipping an exon in the middle. Not a lot. The arc count suggests that most of the isoforms go to the typical pattern, and it's more rare, but it's an event that does occur. And I can see that as I look through the different annotations, I can see, well, there's a splice form right here that lacks this isoform X3 that lacks this particular exon, right? The second to last exon. So it's a great tool for quickly browsing through, and I'm just clicking on this receive. Now, if I find some that I'm interested in, I can select it and add it to my favorites. And so I'll have a favorites list. And then I also have a recents list, what have I looked at recently. So as I'm navigating, and if I find some that have differential kind of patterns that I think are interesting, I can add them to the favorites. And here I can see, again, I'm going to zoom in a little bit. And this is what really makes this software powerful, is that it's very dynamic and user-friendly, and I can quickly zoom in on areas that are interesting and then scan the annotation to see, are there annotations there that support what I'm seeing in terms of the alignment pattern. And again, I'm just clicking in this list and saying, do I find more? All these genes that met my criteria, do any of them show splice pattern differences between the 2 different tissue types? And so I see here's another one where it looks like there's some kind of splicing event occurring in this region. And so I can load this in. Now this gene has, so I'm looking again up at these annotations, and I'm looking for, are there tick marks? And there I can see the start point. There's a whole subset of isoforms here that have an alternate start site. So, and that's, it looks like in this case in the brain we have those isoforms present, but not in the universal reference. And so it's just kind of an interesting way. So again, this was filtered off of expression, now I'm going and looking to see, well, is there also isoform differences that are present? And I can just kind of work through the software. Now, if I find an area that I want to make an image out of, again, the flexibility that we have here in terms of scaling this image how I want it is powerful. But I can also save the image out now. So I'm going to export the image, and this is the analysis. And I can pick a PDF format, or I can pick something that's new with our applications, Megaline Pro and also GenVision Pro, is the ability to go right to a PowerPoint. And so I can, so if I need to further edit this image, I can export that to, let me just find it there on my desktop. So now it's in PowerPoint, and these are all objects. So if there's something of interest, like maybe it's that arc, I can go in and I can format it and change the style. I think that's how I want to do it. Oh, I just did the box. My bad. There's the arc. And so there, I just highlighted that arc just by changing the color. And I can move it around and edit the text. They're all objects, so if I want to make that, tweak it to get it how I want it for, say, a publication. I can do that in something as simple as PowerPoint or even PDF. So that's one approach to the finding some isoforms. I'm just going to show you one last thing here is another way that we can find isoforms, and that's to go back here to ArrayStar. So we base that first search off of interesting gene expression patterns. Sometimes the gene expression patterns aren't interesting and they're just the same, and the only thing that is interesting are the isoforms. And so you don't know. So what I'm going to do here is I'm going to go to the scatter plot, and I'm just going to select some more highly expressed genes that don't show a dramatic expression change. They're not 8-fold change genes here. Most of them, they're, more or less along the same relative expression between the two. So I might not be able to find these based off expression overall for the gene. They're just kind of in the mix. And you can see there's about 7,000 genes here that are expressed fairly high and are roughly the same between the two different tissues. So I'm going to say, show me the table of isoforms. So what I've done is again, went from the scatter plot, and I just selected a bunch of the genes that don't show dramatic change in overall gene expression. And I said, just open that subset, the isoforms for those genes. So we had this thing called an isoform table in ArrayStar. And when I look at the isoform table, I can see that we have the isoform name and the gene name, so I can see what genes the isoforms came from. And then I can see that there's some RPKM values based on these different isoforms per gene. And so I can look for differences. I can apply a filter, just like we did before for fold change, and then go and look at those particular isoforms to see, if they're interesting. And I did that for a couple. I just want to show you, there's one that I was looking at earlier, and I'm just going to search it up. This CAPZB, in this particular one, I can see that one of the isoforms, isoform 2, shows in brain a much higher level of expression than it does in the reference. And so you'll find some of these, based on the RPKM values that ArrayStar can calculate at the isoform level. And again, as we could before, I can send that selection to GenVision Pro. And at this scale, we can't see what's interesting, but we'll zoom in. And now it just adds that gene, you know, to my list. So here's that particular isoform. And if I look way at the 5' end, we'll move over there a little bit more and zoom in a little bit. There's an interesting pattern right there. And so again, these are genes. This gene is expressed at about the same level in the two tissue types. And I just use the isoform table. And what it's detecting is this this isoform 2. If I hover over it, you can say it's got this exon that defines that isoform. That makes it different from isoform 1, which doesn't have that exon. So we're detecting that in a ray star, and we could see that it occurred in the brain and not the UHR by the fold change for that particular isoform. So just another way, you know, if what's interesting is in your gene expression patterns is isoform and really doesn't impact the overall expression. As you can see, these coverage peaks are about the same. You can use that isoform table to also find differentially expressed isoforms between your experiments and get a very nice picture and visual confirmation of the isoform table.